Hello my dear students, a very warm welcome again to your Power Hour sessions of Zoology. I am Dr. Ami Bayani welcoming you back on these Power Hour of Zoology sessions. So today we are going to continue with the Unit 5 that we had started in the last class, and we had completed 3 chapters. I hope you have studied them well and all your doubts that were there are now crystal clear. So today we are moving ahead with another session and covering up important topics like we did last time of the chapters excretory products and its elimination, neural control and coordination, and chemical coordination and integration. So we are first going to start with excretory products and its elimination. Now when we talk about excretory products, what are we going to cover up in this chapter or in this lecture? We are going to cover up the physiology of how urine formation takes place, and we are going to talk about osmoregulation or regulation of kidney functions, because these are some of the tricky topics that students find difficult. So not to worry, I am here for you all now, and we will be covering these sessions, making them absolutely crystal clear. Neural control and coordination, we are going to talk about nerve impulse conduction. We are also going to have a small glimpse of synapse into it. And in chemical coordination, we are going to talk about mechanism of hormone action. We are going to take 2 important glands from where large number of questions come, that is hypothalamus associated with pituitary and thyroid gland. So let's get started. Now, we all know that there are a lot of metabolic activities occurring in the body. Now, these metabolic activities, when are happening in the body, will lead to formation of some byproducts. Some are useful, which the body uses, but however, some are not useful and they are waste products which need to be eliminated out of the body. Now, one of the important nitrogenous waste that has to be excreted out of the body is urea. How is urea excreted? Through urine. And where is urine produced? Urine is produced in the kidneys. So how is it produced is what we are going to discuss today. Now, when we talk about urine production which takes place in the kidneys, we know that the basic functional unit of the kidney is what? Is nothing but nephron. You are well aware of the structure of nephron, so we will directly go on to understanding of the functioning, that is how urine formation takes place. Now, along with urine formation, I am also going to incorporate the functioning of different parts of the nephron. So, the process of urine formation, now this itself can come as our MCQ. That process of urine formation occurs how? So, this can occur in 3 steps. What are the 3 steps? The 3 steps that we are going to discuss is 1, glomerular filtration, which is also sometimes called as, can you recall what is it also called as? Ultrafiltration. Why? We will discuss. Reabsorption and tubular secretion. All the 3 steps very, very, very important. Now, where does this glomerular filtration take place? As the name suggests, the first step, that is glomerular filtration, takes place in the glomerulus. Where does it take place? In the glomerulus. The second and the third step, that is reabsorption and tubular secretion, you can see itself the name says tubular secretion. So reabsorption and tubular secretion takes place in the renal tubules. Again, this can come as your MCQ, so be very, very careful and remember it. Now let us go on to understanding of the first step, that is glomerular filtration. Now, glomerular filtration, commonly it is said that kidneys are the filters of the body which remove the waste from the blood. Now, how does this glomerular filtration take place? When we are talking about glomerular filtration, there is a particular amount of blood that goes to the kidneys. How much is the amount of blood that goes to the kidneys? Aorta has its branch called renal artery, which takes approximately 1100 to 1200 ml of blood to both the kidneys every minute. That is practically, you have to remember, is 1/5 of the total cardiac output. That is 1/5 of the amount of blood that is pumped by each ventricle per minute. So out of the 5 liters of blood that the heart pumps out, 1/5 of the blood goes to the kidneys every minute. Important MCQ, please make a note of it. And when it enters into the kidneys, it will pass into the glomerulus. How will it go into the glomerulus? Through the blood vessel called afferent arteriole. Once the blood enters through the afferent arteriole, it will enter into the tuft of capillaries which is present here, and this tuft of capillaries is called as glomerulus. Now in the glomerulus, if you see, now these are the basically the glomerular epithelial cells. There are minute spaces which are present. You can see these small dots which are there. Once blood enters, there are these minute small pore-like structures which are present. That is spaces between the cells of the glomerulus, that is the endothelium of the glomerulus, through which the blood is filtered out. And there is also something called as the the membrane which is formed, which is called as the filtration membrane. Now if I draw it for you here, see, if this is the Bowman's capsule, and here we have the glomerulus. So once the blood enters through this afferent arteriole, there are small spaces which are present between the endothelial cells which are called as small fenestrae. Now this is not given in the NCERT, if you can remember, well and good. Now these fenestrae will allow large amount of substances to move out. But how will they move out? They will move out because of a pressure that is created. If you see here, the diameter of the afferent arteriole is much broader as compared to the diameter of the efferent arteriole. So the amount of blood that enters into the glomerulus per minute is not equal to the amount of blood that is leaving. So if there is large amount of blood entering as compared to small amount of blood that is leaving out, there will be pressure that will be created here into the glomerulus, and that pressure is called as glomerular capillary blood pressure. What is it called? Glomerular capillary blood pressure. My dear students, remember, this can again come as your MCQ, that what is it that brings about the filtration? It is this pressure, which is glomerular capillary blood pressure, created because the afferent arteriole is broader as compared to the efferent arteriole, creating that blood pressure here. Now because there are fenestrae, the small substances will start moving out. Now what will move out? Everything practically that is present in the plasma. Okay, so RBCs, WBCs and platelets are not going to come out. It is the plasma, and in the plasma also proteins are not going to come out. Now they will move out from here, but what about the Bowman's capsule? There are specialized epithelial cells here lining the Bowman's capsule, which are called as podocytes. Now if you can see here, this is one cell. Let me use this color for y'all. This is one cell. Now can you see there are these finger-like projections which are present here. Okay, this is one cell. Now here there is one more cell. See? So these are like interdigitations, okay. So there are small slits which are present between the podocytes, and these slits are called as filtration slits. What are they called? Filtration slits. So through the fenestrae, when substances move out, they will pass through these filtration slits and now enter into the space present here. And practically, as I told you, large amount of substances will move out, practically everything that is there in the plasma except protein. So what comes out as the filtrate is called as deproteinized plasma. What do we call it as? Deproteinized plasma. So what, through where is the filtration taking place? MCQ, it is taking place through the glomerulus. What causes the filtration? The glomerular capillary blood pressure. And through which membrane does it come out? Is the filtration membrane. Now what is this filtration membrane made up of? I just explained you. I am again enumerating. There are three things that the filtration membrane is made up of. One is the endothelium of the glomerular blood vessels. Second is the epithelium of the Bowman's capsule. So endothelium, where there are these fenestrae present. The epithelium, which is made up of specialized cells called podocytes, and the basement membrane between the two. Like we had studied in respiration where diffusion membrane is formed, similarly here filtration membrane is formed. Is this much clear? Okay. Moving further ahead. So as I told you, 1100 to 1200 ml of blood is being filtered every minute. Now so much amount of blood is filtered. How much is the filtrate formed because of the pressure? The filtrate formed is mind-boggling, is approximately 125 ml per minute, which is 180 liters per day. 180 liters per day. Practically, I told you everything that is present in the plasma, water, solutes, ions, waste products, some micronutrients, nutrients which are monomers in the form of monomers, all of that is being filtered out, producing 180 liters of filtrate, that is 180 liters per day, that is 125 ml per minute. Now, if you think, can we afford to lose 180 liters of filtrate every day? My dear children, we have 5 liters of blood. If everything gets filtered out, we'll practically not be able to survive even for 24 hours, isn't it? So, is it important or essential to remove all of this? No, there are some vital essential substances that are also filtered out. But we will have to reabsorb them back. Why? Because they are important for the body. Water, important for the body. And if you see, the amount of urine that is formed every day is just 1.5 liters, 1 to 1.5 liters. However, this is normal. Depending upon multiple factors, it will vary. So, if we just take an average of 1.5 liters per day urine formed and 180 liters of filtrate formed, 99% will be reabsorbed. Very good. So, how will the reabsorption take place and from where will the reabsorption take place? Let us first understand what do we mean by the word reabsorption and what do we mean by the word secretion. That is the next 2 steps of urine formation. When we are talking about reabsorption, it is anything that goes from the tubules into the blood. We know the nephrons are surrounded by peritubular capillaries, one of the branch that surrounds the loop of Henle. Can you quickly tell me what is that branch? Yes, the branch that surrounds it is called as vasa recta. Yes, so anything that is going from the tubules into the blood is called reabsorption. And when we are talking about vice versa, that is from the blood, anything that is going into the tubules is called as secretion. Are you understanding? Remember this, okay? Now, this is the understanding of reabsorption and secretion. But how are things reabsorbed? Reabsorption can take place either by active transport or by passive transport. Now, what are things that are reabsorbed actively? Glucose, sodium, amino acids, etc. These are the substances that are reabsorbed actively. Whereas, nitrogenous waste. Now, you will say is nitrogenous waste. For removal of nitrogenous waste, the entire process takes place. Do we need nitrogenous waste? Yes. Countercurrent mechanism, you will study nitrogenous waste are important in small amount, which has to be present to create an osmolarity, which is desired in the medullary interstitium for concentration of urine. So nitrogenous waste passively diffuse, small amount, and water. But remember, my dear children, passive movement of water is in the initial segments of the nephron. Is that clear? So let us move ahead to understanding. Now this is the nephron which we opened up to understand. So we have a Malpighian corpuscle here. We know that the filtration has taken place at the level of glomerulus. Now the filtrate passes through the PCT. PCT is lined by brush-bordered cuboidal epithelium. Now tell me quickly what is the function of brush-border epithelium? Hello my dear bachas, yeah, hi everyone. Quickly tell me, what is the function of the brush border epithelium? Yes, I am waiting for your answers. Yes, very good. Brush border epithelium increases the surface area. So, because there is 180 liters filtrate formed every day and 99% has to be reabsorbed, main reabsorption takes place from the PCT. What is reabsorbed from the PCT? We have 70 to 80% of electrolytes that are reabsorbed and equal amount of water that is reabsorbed. 70 to 80% electrolytes, 70 to 80% water, and almost all nutrients are also reabsorbed from the PCT. Once this takes place, then comes the loop of Henle. From the descending limb of loop of Henle, it is permeable to water, so water is moved, is reabsorbed. When you are talking about the ascending limb of loop of Henle, ascending limb of loop of Henle is the place where there is minimum reabsorption taking place. It is this part which is permeable to ions, impermeable to water. Descending limb is permeable to water, impermeable to ions. Vice versa is in the ascending limb of loop of Henle. So you see, the ions are pumped into the medullary interstitium. This loop of Henle mainly helps in concentration of urine. This has come as your MCQ. Then the filtrate goes into the DCT. In the DCT, there is conditional reabsorption taking place of sodium and water. What is conditional reabsorption? Now, 70 to 80% water that is reabsorbed from the PCT is a compulsory reabsorption, otherwise the person will die. However, depending upon the condition of the body, whether the person is dehydrated or not, accordingly sodium and water will be reabsorbed, which we will see in some time now. And at the same time, there is small amount of bicarbonate ions also that are reabsorbed. You can see here. Now these bicarbonates that are reabsorbed along with the secretion that takes place of hydrogen, of ammonia from the PCT, of hydrogen ions, potassium and ammonia from the DCT will help in maintaining the pH and the ionic balance of the body. So once everything is reabsorbed, whatever is not required is secreted back into the tubules. Then what comes into the collecting duct is the one that will be excreted out. However, from the collecting duct, small amount of water will also be reabsorbed. And there will be minute amount of urea that will go into the interstitium, which will be pumped back into the ascending limb of loop of Henle. So after filtration, after whatever is required by the body is reabsorbed, whatever is not required is secreted back, then what is coming out is what is urine. So this is the functioning of different parts of the nephron, and this is how urine formation takes place. However, the functioning of kidney is not just urine formation. Then what is the other functioning of the kidney is to maintain osmoregulation. Osmoregulation is maintaining salt and water balance in the body. How does it do that? It does that with the help of hypothalamus, JGA's juxtaglomerular apparatus and heart. We will see one after the other. Let us understand how the regulation takes place through the pituitary gland and through the hypothalamus. Now there are hypothalamus we will be studying in some time. Now I will also explain you here. Hypothalamus is the basal part of the diencephalon. Diencephalon is a part of our forebrain. Descending down or hanging down from the diencephalon is a small gland which is called as pituitary gland. Anterior lobe of pituitary, intermediate lobe of pituitary and posterior. There are three lobes of pituitary. We have the anterior lobe, the intermediate lobe and we have the posterior lobe of pituitary. The anterior lobe of pituitary is connected to the hypothalamus or is controlled by the hypothalamus via specialized portal circulation. Posterior pituitary is the part which does not secrete any hormone. It just stores and releases the hormone that is produced by the hypothalamic neurons. And one of them is ADH, which plays a vital role in maintaining our osmolarity. Now let us understand ADH is anti-diuretic hormone. hormone which is anti meaning against, diuresis. Diuresis is excessive urination. Hormone that prevents excessive urination is called as antidiuretic hormone. The another name for this hormone is vasopressin. What is the other name? Vasopressin. Now how does this function? Now say for example, this poor chap was playing in the sun and did not have water, so he was completely drenched in sweat, dehydrated, and there's decrease in the blood volume because there was loss of water from the body, or there is decrease in the body fluid volume, that is blood volume, thereby changing the ionic concentration. Once the ionic concentration changes, the blood with the changed ionic concentration, when passes through the brain, passes through the hypothalamus, there are specialized receptors called osmoreceptors which are present in the hypothalamus. These osmoreceptors start blinking, red flag, red flag. Once these get stimulated and start blinking, they will send a signal to the posterior pituitary and tell posterior pituitary, neurohypophysis, that is posterior pituitary, I had given you ADH which you had stored, now start releasing ADH. Once it starts releasing ADH, this ADH will now go to the DCT, that is to the later parts of the renal tubule, and from the renal tubule reabsorb water. Once water is reabsorbed, put back into the peritubular capillaries, that is into the blood circulation, the blood volume increases. Blood volume increases, it will now send a negative feedback and tell the osmoreceptors, stop. Now the amount of blood is again coming back to normal. But ADH, as I told you, the other name is vasopressin. Vasopressin brings about vasoconstriction. So another function of ADH is vasoconstriction. You see, this is the normal lumen of the normal artery. ADH will bring about constriction, reducing the lumen. As the lumen reduces, the pressure increases. As the pressure increases, the blood pressure comes back to normal, GFR coming back to normal. That is how when there is dehydration, ADH will help again regain back the blood volume and thereby the ionic concentration. This is one mechanism. The second mechanism is RAAS mechanism, that is renin angiotensin aldosterone mechanism, or we can also say that this is the mechanism with the help of JGA, juxtaglomerular apparatus. Now let us understand what is this juxtaglomerular apparatus. Give me a second, please. I hope you all are listening carefully. Yes. Okay. Now what is this juxtaglomerular apparatus? Now we know that there are approximately 1 million of nephrons which are present in a kidney. Yes. Now they are all compactly arranged. They are not arranged in the manner that we had seen here. Opening their arms like this and sitting there. Okay. They are all in a very compact arrangement situated into the kidneys. So if this is our Bowman's capsule and glomerulus, our PCT, we have our loop of Henle and DCT. You see, now there is a part of the nephron or of the DCT which is very close to the glomerulus. If I just expand this much portion and show you, this is how it appears. We have just taken a small section of the DCT and we have enlarged it here. So you see, this is the DCT section. This is the efferent arteriole. This is the efferent arteriole. Now, there is a part of the DCT which is close to the glomerulus, right? So this part has cells which are modified. You see, the cells here are cuboidal. However, the cells here are slightly modified, and we call these cells as macula densa. What are we calling them? Macula densa. Similarly, afferent arteriole has slight cells. It's an arteriole, so it will have tunica media, circulation chapter. Media will have muscles. Some of the muscle cells are modified to form what is called as juxtaglomerular cells. Now, these juxtaglomerular cells are the ones that will secrete what is called as renin. They secrete renin. Okay, and these macula densa cells are chemosensitive. They are chemosensitive, acting as chemoreceptors. Now, if there is change in the blood volume, if there is change in the GFR, if the blood volume changes, the blood pressure changes, the glomerular blood flow changes, and the GFR will also be affected. Now, if there is change in the blood volume in such a way that it reduces, like we saw in ADH, here also what will happen is that the amount of filtrate that has to be formed, or the type of filtrate that has to be formed, will be affected. If the type of filtrate formed will be affected, the change filtrate, when it passes through the DCT, these cells which are there of the macula densa will get stimulated. Once these get stimulated, because they are chemoreceptors, they will detect change in the chemical composition. Once they get stimulated, they will send signal to the juxtaglomerular cells of the afferent arteriole. The juxtaglomerular cells will now secrete renin. This renin will act on an enzyme which is present in the blood but in an inactive form called angiotensinogen. Now, angiotensinogen will be activated by renin. Angiotensinogen will be converted into angiotensin 1, eventually into angiotensin 2. Angiotensin. Angio, blood vessels, tensin, tension. It again creates tension on the blood vessels, narrowing down the blood vessels, thereby constricting the blood vessels. Condition called extremely sorry. Vasoconstriction. As vasoconstriction takes place, the blood pressure increases, glomerular blood pressure increases, and the GFR is brought back to normal. But that is one method. What does the name say? It is through renin-angiotensin-aldosterone system. Renin we studied, angiotensin we studied. What is this aldosterone? Now, once the angiotensin 2 is formed, angiotensin 2 goes to the adrenal gland and tells the adrenal gland, cortex, please secrete aldosterone. Once aldosterone is secreted, aldosterone goes to the distal part of the renal tubules. I just explained, the distal part brings about conditional reabsorption. So, from the distal part of the renal tubule, sodium is reabsorbed. Once sodium is reabsorbed, water will move in by osmosis. As sodium and water is moving in through osmosis, sodium by active transport, the blood volume increases. As the blood volume increases, the blood pressure increases. As the blood pressure increases, the GFR is brought back to normal. Is this much clear? So that is renin, angiotensin, aldosterone system. Is that clear? Cool. Moving ahead to the third mechanism, which is by ANF, that is through heart. Now, what is this ANF? ANF stands for atrial natriuretic factor, the factor that brings about natriuresis. We saw two mechanisms where we saw decrease in the blood volume. Decrease in the blood volume was stimulating ADH secretion. Decrease in the blood volume was also now bringing about the secretion or stimulation of RAAS mechanism, thereby secretion of aldosterone, thereby increasing the blood volume. Now, these were the two mechanisms which were taking place because of decrease in the blood volume and the blood pressure. Now, what if the blood volume increases? What will happen? Say, for example, the person has had large amount of fluid that day. Now, you tell me, if you have large amount of fluid a particular day, say 4 liters, 5 liters, what is going to happen? you will have the urge to pass urination again and again. Why? Because the blood volume has increased, and body, to maintain homeostasis, will have to reduce the blood volume, thereby increase the urine output. Isn't it? How does it do that? When the blood volume increases, there's large amount of blood going to the heart. Now the atrial walls will secrete a factor called atrial natriuretic factor, which brings about secretion or excretion of sodium in the urine. Once sodium is put into the urine, water will move behind it and there will be loss of water. Now it also brings about vasodilation. Remember, RAAS mechanism was bringing about vasoconstriction. ADH was bringing about vasoconstriction. ANF is bringing about vasodilation. Once there is vasodilation, you see, this is the normal artery and this is the increased diameter of the artery. Here the diameter is only this much, whereas here the diameter has increased. Vasodilation, thereby decreasing the blood pressure. Now RAAS was bringing about vasoconstriction. This is bringing about vasodilation. So ANF acts as a check on RAAS mechanism, and this can come as our MCQ. Important MCQ. Please make a note of it and remember it carefully. So I hope that the osmoregulation part is also clear. The functioning of the renal tubules, the part of the nephrons is also clear, and urine formation is also clear. So quickly let us move ahead to answer questions and see some of the questions. Now again, as I do always, I have brought about your assertion reason. Now when we are talking about assertion, aldosterone causes reabsorption of sodium and water from the distal parts of the renal tubule, and reason is an increase in the blood flow to the atria of the heart can cause release of ANF. Now quickly tell me the answer for this. I am giving you 30 seconds. 30 seconds, your time starts now. Quick answers. Okay, Taksh has already started to answer. Very good, Taksh. Mrunal, very good. Khushi, Soni, Harshali, waiting for your answers, dear. Khushi, very good. Okay, let's see. Aldosterone causes reabsorption of sodium and water from the distal part of the renal tubules. Yes, yes, yes. So this is a true statement. Increase in the blood flow to the atria of the heart can cause release of ANF. Yes, this is again true. But is this the reason for assertion? No. So all of you all very good, have answered it correctly. The answer key is B. Both assertion and reason are true, but reason is not the correct explanation. Very good, proud of my children. Very good. Chalo. Let's move on to the next question. Again an assertion, reason. Assertion. Angiotensin of the RAAS mechanism and ANF are antagonistic to each other. Reason is 99% of the ultrafiltrate is formed is reabsorbed from the renal tubules. Quickly, 30 seconds again. Yes. Taksh, very good again. First to answer, Taksh, very good. Very good, Mrinal. Yes, so let's see. Angiotensin of RAAS mechanism and ANF are antagonistic. Yes, it's a true statement. 99% of the ultrafiltrate formed is reabsorbed by the renal tubules. Again, yes, it is absolutely correct. But is it the correct answer or the reason? No, no, no. So both assertion, reason true, and reason is not the correct explanation. Very good, Khushi. Very good, Taksh. Very good, Mrinal. Very good, Harshali. Very good. Chaliye. One more question quickly. Match the column. Thirty seconds. Come on, let's do it in 15 seconds. Can you do it in 15 seconds? Chalo. I know you can do it. Fast, 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 my dear bachas. Yes, Khushi, very good. Mrunal, very good. Daksh, very nice. Okay, let's see. Ultrafiltration, where does ultrafiltration take place? Ultrafiltration is by the glomerulus, so 4. Concentration of urine, I just told you, is Henle's loop, so 3. Conditional reabsorption is DCT, 2. And reabsorption of 70 to 80% electrolytes, almost all nutrients, is 1. 4, 3, 2, 1, let us see. 4, 4, 4, 4, 4. From the first option itself, we will know the answer. Very good, very good, very good, very good. All of you are giving correct answers. Proud of you all. Very good. So that was about the excretion chapter. Moving ahead to understanding of another core concept of neural system, that is action potential, that is nerve impulse conduction. Now listen carefully, it's very, very easy, okay? Now when we are talking about nerve impulse, you can see these are the impulses that are being conducted. They are like electric currents. They are nothing but what is called as action potential. What are they? They are nothing but action potential. Now why is it that the neuron is capable of conducting, generating its impulse and conducting its impulse? This can come as your assertion reason. It is because, can you tell me quickly, why is it because? Anyone? It is because the neurons are into a polarized condition. Now what do we mean by the word polarized condition? Yes, very good children. Yes. Now what is this polarized condition? Now polarized condition means, if you are talking about this as a neuron, the outside has electropositive charge, the inside has electronegative charge. Outside is electropositive, inside is electronegative when it is in a resting state. Now what keeps it in a positive and what keeps the inside the negative? There are 3 conditions. Now when we are talking about neuron, it's a cell, right? And you've studied in the cell chapter that cell membrane is selectively permeable. The outside of the neuron, that is the extracellular fluid, has large amount of sodium ions into it, and the intracellular fluid has large amount of potassium. Now the permeability is such that it allows the potassium influx but does not allow the sodium, it allows the potassium efflux but does not allow the sodium influx. So large amount of potassium will move out but negligible amount of sodium enters inside. If cations are moving outside but not allowed to move inside, outside becomes electropositive, inside becomes electronegative. One reason. Second, there are specialized pumps called sodium-potassium ATPase pumps. Now these sodium-potassium ATPase pumps will cause efflux, that is moving of 3 sodium outside to 2 potassium inside. When 3 sodium moves outside and 2 potassium moves inside, one pump using ATP once will cause a net positive charge outside and a net negative charge inside, creating an electropositivity outside and an electronegativity inside. Third, there are negatively charged proteins. Now, in the first lecture of the power hour, we studied proteins. I told you they behave differently in pH of different solution. So, here they behave as negatively charged ions, and these proteins which are there, because their macromolecules cannot move out, so they remain inside, creating a negativity inside. Thereby, outside is electropositive, inside is electronegative. There are some specialized ion channels which are called for sodium and which are for potassium. They are specific for these ions, which will work and allow only these ions to move either inside or outside. Which ones? How? We'll see. So thereby it is in a polarized state. Now whenever there is an impulse that comes, this impulse will cause change in the permeability. This stimulus causes change in the permeability. How does it change? It allows large amount of sodium to have an influx. When sodium has an influx, there will be large amount of sodium ions entering inside, making the inside or changing the inside electropositive. At this point of time, the sodium channels open up, and through them also large amount of sodium moves inside. Thereby inside starts to turn electropositive and pump stops. As inside turns electropositive, outside turns electronegative, the polarized condition is reversed. You call it as depolarized condition. This generates a small electric current here. This small electric current is action potential, is nothing but nerve impulse. Is this much clear? Yes. Once the nerve impulse is generated, say for example, we are talking about this site A. This is site A, this is site B, site C, and so on and so forth. Now, if you are talking about site A has been depolarized, right? You can see that inside is electropositive, outside is electronegative. See, inside electropositive, outside is electronegative. Reversal of what was in the polarized state. However, if you see the side B is still in the polarized state. Inside is negative, outside is charge electropositive. There is a gradient which is present. Now we know that current, physics, you know that current will always move from a positive charge to a negative charge. So the depolarized condition at the site A now will cause the electric current or the charge to move from A to B on the inside, and will cause it to move from B to A on the outside, completing the circuit and thereby now depolarizing the site which is B, and site A is again going into a repolarization. Repolarization means bringing it back to the polarized condition. Now, I had told you that there is change in the permeability because of stimulus. Now, this stimulus-induced change in the permeability is very short-lived. Short-lived in the sense it's for few fraction of a second, and once the action potential is generated, the permeability again restores back to normal. What was normal? It was allowing efflux of potassium but not influx of sodium. The same thing happens here. Once the impulse which is there goes from site A to site B, that is from site A to site B, in the site A, repolarization takes place where the permeability for sodium stops. So potassium can move out, but sodium influx stops. The pump starts to work, pumping the sodium outside. At the same time, the potassium voltage gated channels open up and pumps the potassium outside, again turning inside electronegative, outside electropositive, bringing it back to the polarized condition, repolarization. That is how nerve impulse generation and conduction takes place. Is that clear? Okay. Let us quickly go on to the answer, to the question. Arrange the following events in the correct order as they occur during the transmission of an action potential along the axon. Depolarization at the point of stimulation, repolarization by efflux of potassium, sodium influx at the point of stimulation, restoration of resting potential, generation of an action potential at the next side. What will be the correct sequence? Okay, dear children, quickly. Yeah. So let us see. What will be the correct sequence? The correct sequence first will be, there will be restoration, sodium influx, there will be sodium influx at the point of stimulation. C will be the first one. Okay, so you have two places where C is the first one. Once sodium influx takes place, that causes depolarization at the point of stimulation. This will now cause generation of the action potential. This will now lead to repolarization by efflux of the potassium and restoration of resting. So we have C, we have A, we have E, CAE, CAE, BD, and that is our answer. Is that clear, everyone? Yes. Moving ahead, yes, very good. Moving ahead to understanding of synapse. Now, impulse has been generated, impulse has been conducted across an axon. So, how it goes from one neuron to another neuron is via synapse. Now, synapses basically, we will see, are of two types. There is a chemical synapse, there is an electrical synapse. Synapse is basically the point of contact between the two neurons. How? There is one neuron which is the presynaptic neuron. There is another neuron which is the postsynaptic neuron. The nerve ending of the presynaptic neuron and the axon or the dendron of the postsynaptic neuron. Now, once the impulse reaches the axon terminal, there are small circular vesicles which are present in these nerve endings, which have chemicals which are called as neurotransmitters. Now once the impulse reaches the nerve endings, these vesicles will start to move towards the membrane. Once it moves towards the membrane, it will now fuse with the membrane, the vesicle membrane, and the nerve the nerve ending membrane will now fuse, and the neurotransmitters will be now released into the gap which is called as synaptic cleft. Once it reaches the synaptic cleft, there is another neuron dendron which is present here, which has receptors for these neurotransmitters. So neurotransmitters binds to these receptors, bringing about depolarization of this another neuron, and the impulse has now gone from one neuron to another neuron. Is that clear? So what are the parts of the synapse? The parts of the synapse are the presynaptic neuron. This is the postsynaptic neuron. There is a gap which is present, which is called as a synaptic cleft. Now this synaptic cleft may or may not be present. Remember, NCERT says that the synapse comprises of a presynaptic neuron, postsynaptic neuron, and the synaptic cleft may or may not be present. Now, what we just now spoke is the chemical synapses. There's one more type of synapse which is called electrical. Why is this called chemical? Because the neurotransmitters are the chemicals which are involved here. When we are talking about the electrical synapse, we have a presynaptic nerve ending. We have the this is the presynaptic nerve ending. This is the postsynaptic neurons dendron. Now if you see here, look at the gap here. This is a synaptic cleft which is a little wider. Whereas if you see here, there's a negligible amount of space which is present here. Now, this space which is there will not act as a hindrance, and once the impulse reaches the presynaptic nerve ending, it will immediately jump to the postsynaptic neuron. So it is like current moving from one neuron to another neuron, and an action potential going from one neuron to another neuron, very similar to the impulse being conducted across a single axon. This is an NCERT statement, remember, can come as your MCQ. So what do you think, which one will be faster, the chemical or the electrical? Obviously, the electrical synapse will be faster as compared to chemical synapse. It will be very quick to transmit the impulses. But we don't need this quick an impulse. Whatever impulses we have, even if it's a reflex action, will utilize the chemical. So you imagine the electrical would be so very fast. So this chemical synapses are much more in abundance in our body, whereas the electrical synapses are rare in our body. However, the impulse conduction is faster in electrical compared to the ones which are in chemical. Is that clear? Very good. Let's quickly go on to an answer. Yes, very good, Taksh. Read the following statements and find out the incorrect ones from the options that are given below. Synapse is formed by the membrane of a presynaptic and a postsynaptic neuron. Synapse is always separated by a synaptic cleft. Impulse transmission across an electrical synapse is always faster than chemical, and electrical synapses are rare in our body. Now quickly tell me which is true and which is false. Very good, Mrunal. Very good. Very quick, fast. Very good. Taksh, Khushi, Harshali. Khushi, very good. Very good. So what is the answer? The answer will be, which is the incorrect one? Synapse is always separated. No, I just told you it may or may not be separated. So this is the answer. So that is about neural. Very good, Taksh. So that is about neural control. Moving ahead to understanding of the chemical control. Now taking a topic where students find a little difficulty. That is how hormones act. Now we are aware that there are two types of hormones which are there in our body. One which is a steroid hormone, one which is a protein hormone. Hormone will always work via bind to its target organ. And when it works on the target organ, how does it work on the target organ? It will always bind to specific receptors which are either on the target organ or within the target organ. Now, what do we mean by that? Let's have a look at it. Now, if you are talking about hormones which are, say, protein hormones. When I am saying protein, I am including peptide, polypeptide and amino acid derivatives here. They have receptors which are present on the cell membrane. Once the hormone comes and binds to the receptor which is present on the cell membrane, it forms a hormone receptor complex. Now this hormone receptor complex generates a secondary messenger. Now we know hormones themselves are chemical messengers. They will now generate second messengers such as we have cyclic AMP, calcium ions, and NCRT also in theory says IP3 inositol triphosphate. This second messenger will now activate certain proteins and will bring about biochemical responses which brings about metabolic changes and physiological effect, such as if you are taking an example of FSH, FSH will act and bring about ovarian growth. Similarly, if you are talking about steroid hormones, when you are talking about steroid hormones, say for example, we are talking about estrogen. Now, steroid hormones will pass through the cell membrane, enter into the cytoplasm. They have specialized receptors which are present within the nucleus. So, it will enter inside into the nucleus and bind with its receptor, forming a hormone receptor complex. Once it forms the hormone receptor complex, it acts on the part of a genome which will now bring about transcription, forming mRNA molecule. You studied it in botany. Once mRNA is formed, it will bring about protein synthesis. These proteins will now act and bring about metabolic changes, physiological responses, and thereby biochemical changes and physiological responses which are the functioning or which are visible to the body. That is how these hormones will work. This can come as your MCQ. These are 19.5 figure in your NCRT. Read them carefully. Let us quickly take a question on this. Identify the chemical nature of hormones with correct examples. Now, if you see here, the hormone is binding to the cell membrane. So, what kind of a hormone will it be? Quick. It will be a steroid hormone or a protein hormone? Will be a steroid or a protein hormone? Yes, it will be a very good protein hormone. So it will be a protein hormone. Now is testosterone a protein hormone? It's a steroid hormone. So it is FSH. So the answer is protein hormone and FSH. Quickly let us take the next question. FSH, the hormone, binds to the pars distalis region of the pituitary, acts on the ovarian cell, produced by the pars distalis, acts on the ovarian cells. Arrange the steps in the correct order. Hormone binds to its receptors on the cell membrane. Physiological responses such as ovarian growth occurs. Biochemical responses within the cells are initiated. Second messenger like cyclic AMP is generated. What is the correct answer? Yeah. Yes, very good Mrinal, very good Taksh. Yeah, let's see. Yes, Taksh, very good. So hormone binds to its receptors on the cell membrane. Yes, that will be the first one. So anywhere where A is the first one, only A answer key has the first option, that is starting with A. Second is that physiological biochemical second messengers are stimulated. Yes, this will bring about biochemical reaction and thereby physiological changes such as ovarian growth. So it will be A, D, C, B. A, D, C, B, and our answer is A. Is that clear, everyone? Yes, very good, Taksh. Okay. Now, we've studied that hormones are basically non-nutrient chemicals which act as intercellular messengers, and we just saw that how they work. Let us, when we are talking about these hormones, are these hormones only secreted by the endocrine glands, my dear children? We've studied earlier in school, we studied that yes, it was only endocrine glands, but now we know it's not just the endocrine glands and non-endocrine tissues also. However, we have multiple endocrine glands that will produce hormones, of which we are taking into consideration the hypothalamus and the pituitary. I have just explained you in excretion chapter, hypothalamus is the base of diencephalon, which is basically a coordinator between the neural and the endocrine function, and a downward projection from it is the pituitary gland. Pituitary gland, I told you, has three parts. It has the anterior lobe, the intermediate lobe, and the posterior lobe. Now, what are the hormones produced by the anterior lobe? Anterior lobe, also called as the pars distalis, and the intermediate lobe, pars intermedia, together called as adenohypophysis. The posterior pituitary is neurohypophysis. Now, if you are talking about the anterior pituitary, it produces a hormone which is called as growth hormone. Now, as the name suggests, it brings about growth of the body, the linear growth of the body. Now, this, in case if there is excess of growth hormone in childhood, it produces gigantism. If there is deficiency, you can see it produces dwarfism. However, in adults, if there is an excess, it produces a deformity which is basically seen first in the facial bones, and that is called as acromegaly. It affects the other parts of the body also, however, first seen in the facial bones. If not treated, it can lead to premature death, so treatment is very, very important. It also secretes another hormone called prolactin. Prolactin acts on the mammary gland, brings about growth of the mammary gland and also milk production. It secretes another hormone called TSH, thyroid stimulating hormone. It goes to the thyroid gland, stimulates the thyroid gland and tells it to produce hormone T3, T4 and TCT, which we'll see in some time. It also secretes another hormone which is ACTH, adrenocorticotrophic hormone, which goes to the adrenal cortex. Now remember, it brings about synthesis and secretion of only glucocorticoids, not all the cortical hormones. It also secretes a hormone called FSH. FSH is follicle stimulating hormone. Now this follicle stimulating hormone in males along with androgen brings about sperm formation, that is spermatogenesis. In females, it brings about growth and development of the ovarian follicles, details of which we will study in reproduction chapter. LH will bring about ovulation, that is release of ovum, and also helps in maintaining corpus luteum in females, whereas LH in males will act on the Leydig cells and bring about synthesis and secretion of androgens. Then comes the intermediate lobe of pituitary. This intermediate lobe of pituitary, though not much functional in the human beings, is functional in lower animals. It produces a hormone called MSH, melanocyte stimulating hormone, and this will regulate the pigmentation of the skin because it acts on the cells called melanocytes. Then the neurohypophysis. I told you this just stores and releases the hormone, does not synthesize the hormone. There are two hormones, one is oxytocin. Now oxytocin acts on the smooth muscles of the body, brings about its contraction. Whereas, in females, it also acts on the uterus and brings about contractions of the uterus, bringing about parturition or helping in parturition, hence called as birth hormone. It also acts on the mammary gland for release, that is secretion of milk. It also produces vasopressin or ADH, which we've already studied in kidney chapter in the beginning of the lecture, which acts and prevents acts on the DCT and the distal part of the nephrons and prevents diuresis. However, its deficiency leads to a condition called diabetes insipidus, wherein there is excessive thirst and excessive urination. So that was about pituitary and hypothalamus. And now moving ahead to the next, which is thyroid, another important gland. It is the largest gland of the body, or the largest endocrine gland. Largest gland, if they ask you, is liver. Largest endocrine gland is the thyroid gland. Where is it situated? Everybody knows it's in the neck region, but where exactly in the neck region? It has two lobes which are on either side of the trachea. This is a trachea, so it's on the either side of the trachea. The two lobes which are there are joined together by isthmus. Now, my dear children, remember, is isthmus glandular in nature? This can come as your true and false statement as a tricky question. Is it glandular? Quickly, can I have the answer? It is not glandular. Very good. It is connective tissue. It is made up of connective tissue and it's called as isthmus. It produces three hormones: thyroxine or tetraiodothyronine, that's T4, and triiodothyronine, that's T3. Both of them have similar functions. We'll talk about it in some time now. And TCT, that is thyrocalcitonin. When we are talking about T3 and T4, they are said to be the main metabolic hormones regulating a basal metabolic rate, thereby regulating the carbohydrate, protein, and lipid or fat metabolism. It brings about physical, mental, and sexual development and helps in RBC production, water, and electrolyte balance. But if you are talking about TCT, thyrocalcitonin, as the name suggests, it regulates the calcium levels by lowering the blood calcium level. Now here you also have to remember another antagonistic hormone to it, and another antagonistic hormone is PTH, parathyroid hormone, which we'll study in some time now. If there is hypothyroidism, that is decrease in the amount of thyroid functioning, it could be because of iodine deficiency, as you can see, leading to enlargement of the thyroid gland called goiter. In adult women, it can lead to irregular menstruation. However, this is important. If it occurs during pregnancy, the baby will be affected wherein there is stunted growth called cretinism, low IQ level, mental retardation, deafness, mutism, abnormal skin, and low intelligent quotient. If there is hypersecretion, which can be because of cancers or nodules, it can lead to another condition. This is one more condition. It can be because of nodules, can be because of cancer, or there is another condition called exophthalmic goiter, Graves' disease. You can see here, there is protrusion of the eyeball. The eyes don't blink much. There's increased BMR, there's weight loss, and there's enlargement of the thyroid gland. And coming to the last gland for the day is parathyroid. These are four glands which are situated on or very close to the thyroid gland. You can look at it or observe it from the dorsal side of the body or the dorsal side of the thyroid gland. One pair each is present on each lobe of the thyroid gland. It secretes a hormone called parathyroid hormone. I just now told you it is antagonistic to TCT, that is thyrocalcitonin. Thyrocalcitonin decreases the blood calcium level, parathormone increases the blood calcium level, both working opposite to each other. So I hope this much is clear. Quickly now let's solve this question. Giving you 30 seconds, quickly solve this question. Gigantism, acromegaly, Graves' disease, cretinism, and hyper of which hormone? Yes, my dear children, can I quickly have your answers? Okay, very good. So let's see. Gigantism is hypersecretion of growth hormone in childhood, so 4. Acromegaly is hypersecretion in adult, stage 3. Graves' disease is hypersecretion of thyroid hormones, 2. And cretinism is hyposecretion. So 4, 3, 2, 1. Again, the answer key D. And last question for the day, pituitary gland, thyroid gland, adrenal gland, pancreas, Graves' disease, diabetes, mellitus diabetes, insipidus, and Addison's disease. Now let's see who's the first one to answer and quick to answer. Khushi. Yes, very good Renal, very good Taksh. Very good Mr. Phantom. Let's see. Pituitary gland deficiency or abnormality causing disease. Pituitary gland, my dear children, if you see, will it cause Graves' disease? No, it will be associated with thyroid, है ना? So your adrenal is associated with Addison's disease, pancreas with diabetes mellitus, pituitary diabetes insipidus. How? We just now studied pituitary, posterior lobe of pituitary stores and releases hormone ADH. If there is ADH deficiency, it causes diabetes insipidus. Yes, very good, Purva. Yes, so this will be our 3142. Let's see, 3142. Very good, Whoopi. Our first option only has the answer. So whenever you are solving MCQs, my dear children, always be careful. You can see the first one. If more than one have first option correct, as you can see A and D, either go to the second or go to the last, and you will quickly get your answer. That's a trick. That's a small, kind of trick how you can solve it fast and quick and save your time. But mind it, also go through the other options, check the other options, and only then mark your answers. Haste is always a waste. Remember, you have to be fast, you have to be quick, but never in haste. So that is the tip for the day. So remember that and solve your MCQs wisely. I know you know everything well, just the presence of mind, and just be a little alert at that point of time. Another tip for the day. So follow that, keep studying, keep enjoying. So that's all for today from my side. See you again with the next session. Hope you enjoyed it today. Till then, bye.